Today, we are diving into a massive breakthrough in understanding our own DNA. We're going to be exploring a brand new AI model from Google DeepMind that isn't just, reading the words in our genetic code. It's finally starting to understand the grammar that holds the whole thing together.
So, let me just ask you this to set the stage. What if I told you that 98% of the instruction manual for a human being was basically a complete mystery? It sounds like something out of a sci-fi movie, right? But for decades, that's been the reality for scientists.
That's right, a whopping 98%. This is the part of our genome that doesn't directly make proteins, the building blocks of our bodies. For the longest time, scientists literally called it junk DNA. Well, it turns out this non-coding DNA is anything but junk. It's more like the operating system. It's the regulatory code that tells our genes when to turn on, when to turn off, and how loud to be. And we now know that tiny single-letter typos in this operating system are linked to all sorts of diseases: schizophrenia, heart disease, even many cancers. The problem? Figuring out which of these little typos matter and how they actually cause disease has been a huge, huge headache for biology. Okay, so here's our roadmap for breaking down this incredible new paper. We'll kick things off with the genome's great mystery, then look at the scientific trade-offs that have really held the field back. Then, of course, we'll introduce Google DeepMind's AlphaGenome, see how it works, how it performed, and wrap up with what this all means for the future of genetics.
So, let's dig into this great mystery. What is it really about? Well, it all comes down to understanding the language of our DNA, beyond just the simple words that code for proteins.
Think of it like this. The protein-coding parts of our DNA, those are the nouns and verbs in our genetic instruction manual. But that other 98%, the non-coding DNA, that's all the grammar, the punctuation, the syntax. It's what controls the entire structure and expression of the story. It tells our cells which genes to read, and when, and how much. A single typo in that grammar can change the entire meaning of a sentence and, lead to disease.
You see, for years, any AI model trying to read this complex genetic grammar has been stuck. It's been forced to make a really fundamental and kind of frustrating trade-off. Basically, previous AI models had to choose. On one hand, they could see the big picture, how distant parts of the DNA strand talk to each other, but they could only see it in blurry, low resolution. Or, on the other hand, they could zoom way in to see the superfine single-letter details, but then they'd lose all that crucial context. At the same time, they had to choose be a generalist that could predict a bunch of things just okay, or be a specialist that did one thing really, really well, but was totally blind to everything else.
And this is exactly where AlphaGenome comes in. This is the problem it was built to solve. It's a model that basically says, nope, I'm not going to choose.
The paper itself is called AlphaGenome, Advancing Regulatory Variant Effect Prediction with a Unified DNA Sequence Model. It comes from a big team over at Google DeepMind, with lead authors including Zhika Ovsek, Natasha Latisheva, and Jun Chung.
And this quote from the paper, it really gets right to the heart of the whole thing. Its key novel idea is that AlphaGenome is a single, unified model that refuses to compromise. It reads these long stretches of DNA to get the context, it predicts lots of different genetic activities at the same time, and it does all of that with single-letter or base pair resolution. It's trying to be both a generalist and a specialist, seeing the forest and the trees.
So the big question is, how on earth do they pull that off? Let's pop the hood and see what makes this thing tick.
The best way to think about its architecture is like this. AlphaGenome takes in a massive chunk of DNA, 1 million letters at a time. It then uses this really clever combo of two AI techniques. First, convolutional layers act like a microscope, spotting all the local patterns and motifs. Then, transformer blocks, the same tech behind models like Gemini, act like a telescope, understanding the long-range grammar and how these distant parts of the DNA influence each other. And the way they trained it is also incredibly smart. In step 1, called pre-training, they train a bunch of different teacher models. Each one becomes an expert on a specific part of the genome. Then, in step 2, a process called distillation, a single student model, which is AlphaGenome, learns the combined wisdom from all of those teachers. This is what makes the final model so powerful, but also really robust and efficient.
Okay, so a brilliant design is one thing, but what really matters is performance. So, does this thing actually work?
And the results are, they're pretty stunning.
When they tested its ability to predict the effects of genetic variations, AlphaGenome matched or beat the state-of-the-art on 24 out of 26 different tests. And get this, it didn't just outperform other generalist models, it even went head-to-head with highly specialized models and beat them at their own game, like predicting how genes are cut and pasted together.
Let's make this real for a second. This image shows an amazing real-world example of its power. Researchers have known for a while that certain mutations in the non-coding DNA near a gene called tau1 can cause a type of leukemia. What AlphaGenome did was take the raw DNA sequence and predict the exact chain of events, how that tiny mutation changes the 3D structure of the DNA, which then ramps up the tau1 gene's activity, driving the cancer. And that is a massive leap, from just seeing a correlation to actually understanding the mechanism.
And this ability to connect the sequence to the function is why this paper is such a big deal. It's a genuine leap forward in our ability to read the book of life.
Ultimately, AlphaGenome is like a powerful new Rosetta Stone for the genome. It helps us translate that mysterious 98% of non-coding DNA into a language of biological function that we can finally start to understand. It's a foundational tool that's going to help us decipher the incredibly complex processes written in our DNA.
And the potential applications here are just vast. I mean, imagine being able to diagnose rare diseases better because you understand the exact impact of a patient's unique mutations. We could design new medicines way faster or run massive experiments on the computer, in silico, to test ideas before even touching a test tube. It could even help us build the next generation of AI that doesn't just read DNA, but maybe one day writes it.
And that leaves us with one final, really exciting thought. For the first time, we are truly beginning to read and understand our entire genetic instruction manual, grammar and all. The question is no longer just about what it says, it's about what we're going to do with that knowledge. Now that we can finally read the code, what will we write next?